Good morning, children.
Good morning, ma'am.
We learned about translation, and I had sent some videos in the group. I hope translation process is better or clearer after seeing those videos. Any doubts? Means you can tell me. I'll clarify it. Any doubts are there? If not, we'll proceed with the next topic. That is Lac Operon. We are going to learn about regulation of gene expression, how genes are expressed or how the expression of genes are regulated. And we will also learn about Lac Operon under regulation of gene expression. And you know that genes upon expressing, they will result in the formation of a polypeptide or a protein. And there are different proteins in our body, each with a specific function. So it is very important that the genes get expressed and they form polypeptides or proteins which carry out different functions in our body. And how the expression of genes are regulated, that we are going to learn. Listen carefully. As I've already told you, it's a very, very important topic. Any doubts? Clarify here itself. Don't wait for the topic to be over or the chapter to be over. Gene expression. Once genes are expressed, it will result. Yes. Once the genes are expressed, it will result in the formation of a polypeptide. Polypeptide means protein. So genes need to be.
In the previous topic of translation.
Yes.
Ma'am, am I audible?
In between, your voice is breaking.
Ma'am, in that elongation process, the ribosome moves, right? That part wasn't clear for me.
Okay, now since I've started with this, next period before I begin, I will explain it. Okay, Nandana?
Okay, ma'am.
Next period, beginning itself, I'll clarify your doubt and then I'll proceed. Okay, gene expression, remind me if I forget. Gene expression, once the genes are expressed, it will result in the formation of a polypeptide, and these polypeptides or proteins perform different functions in our body. And the regulation of gene expression, it is taking place at different levels, or the expression of genes are regulated at different levels. You have understood how the genes are getting. Gene means a segment of DNA. And in translation, transcription and all, you have learned how the structural gene is getting transformed, how it is getting transcribed, and how the transcribed segment is getting translated. So once translation is completed, the polypeptides are formed, the gene is getting expressed, resulting in the formation of proteins, and these proteins perform different functions in the body. And the gene expression can be controlled, can be regulated at different levels, at different stages of gene expression. For example, if you take the case of eukaryotes, this can be regulated at 4 levels: transcriptional level, processing level, transport while messenger RNA is being transported, and at the translational level. So with the first one, transcriptional level. What is transcription, Vidar? What is transcription?
Copying of genetic information from one strand of DNA into RNA.
That is transcription. So that means while and once transcription is completed, immediately after transcription, what you get is not messenger RNA. You are getting a primary transcript. What is the other name for primary transcript? What is the other name? Alaviga, what is the other name? The primary transcript. This should undergo modifications and it should form like splicing, capping, tailing. Then it will form messenger RNA. But as soon as it is formed, what do you call it? Rijas. HNRNA or heterogeneous nuclear RNA. And this RNA will undergo changes and will transform into messenger RNA. So while primary transcript is being formed, this can be regulated. The gene expression can be regulated during the formation of primary transcript, or it can be regulated at the processing level when the splicing is carried out. Why splicing is carried out? Why splicing is carried out? What
To remove
for?
introns.
Yes, to remove
Introns.
introns and to join exons. So during that stage, gene expression can be regulated. And I told you, messenger RNA, in that figure also, in the video also, it was clear. Messenger RNA's formation is taking place in the nucleus. Once it is formed, through the nuclear pore, it will move towards the cytoplasm. And translation is taking place in the cytoplasm. Okay. So that during that time, when it is being transported from the nucleus to cytoplasm, the gene expression can be regulated, or finally at the translational level, when it is getting, when the messenger RNA is getting translated or changed into proteins or polypeptides, during that time, the gene can get regulated. Gene expression can be regulated. Children, I'll repeat again. We were learning about regulation of gene expression. Why gene has to be, gene is a segment of DNA. This should be expressed. Upon expressing, polypeptide or proteins are formed. So all the stages from transcription to translation, they are involved in the expression of genes. So genes will be expressed. Upon expression, they will form proteins, and different proteins are there and they are having different functions. And then I told you that the regulation, the gene expression should be controlled or regulated. There is a monitoring system for this gene expression, and this can take place at different levels, either during the formation of primary transcript, or during the splicing process, or while messenger RNA is transported to the cytoplasm from the nucleus, or at the translational level. Anytime this can get regulated in case of eukaryotes. 4 stages where the gene expression is regulated. Apart from that, there are so many other features like metabolic features, physiological or environmental characters can regulate the expression of genes. The gene expression can be regulated by metabolic, physiological and environmental factors too. You know that it is very important that genes get expressed and form proteins. Only then the different functions can be carried out. Proteins, enzymes, hormones, all these are proteins. So only these proteins are produced as a result of the expression of genes. Different activities in your body can take place. So the expression of genes is very, very important. Expression of genes coding for enzymes is required only when the substrate for that enzyme is available. Now, I told you there are different metabolic, environmental, and physiological factors regulating the expression of genes. And enzymes are also proteins. They are also polypeptides. And children, just say, for example, a particular enzyme is needed. And what is the role of an enzyme? Enzyme will act on a substrate and it will speed up the formation of products. Enzyme will speed up the reaction. Enzyme will act on a substrate and convert it into products faster than in the presence. If the same process is taking place in the absence of an enzyme, it might take more time. So when an enzyme is present, the process can be proceeded faster. And when do we need an enzyme? When that substrate is there. If that substrate is not there, we cannot change it into, convert it into products. So then do we need those enzymes? Are you getting the point? A particular enzyme is acting on a particular substrate. Upon acting, children, the enzyme will act on proteins, substrates. Enzyme acts on substrate and they will result in the formation of enzyme substrate complex. Then enzymes plus products. I don't know whether you have learned it in chemistry, or you can say that enzyme will act on the substrate so that it can result in the formation of products. Enzymes are proteins. Enzymes are polypeptides. And if the substrate is not there, do we need enzyme?
Ma'am,
Ah,
Nandana is in meeting.
I have admitted her. See, substrate is not there. Then do we need enzymes? Not required. Enzyme is needed when the substrate is there. If substrates are not there, enzyme has got no function. Are you following what I'm saying? Are you following? Can I get a green signal to proceed?
Yes, ma'am.
Okay. So when when substrates are there, we need enzyme. But when substrate is not there, we don't require enzyme. So then the gene need not get expressed. The gene responsible for the production of this enzyme need not get expressed at that time because we don't need enzyme. Why we don't need enzyme? We don't have a substrate. Only when substrate is there, we need an enzyme. Only then the genes coding for that need to be expressed. That is why it is mentioned there are different factors, metabolic, physiological, environmental factors can regulate the expression of genes. Expression of genes which code for an enzyme is required only when the substrate is available. If that substrate is not there, we don't need that enzyme, so that gene need not get expressed. They have given an example for this. Hydrolysis of lactose in E. coli bacteria, Escherichia coli bacteria. Lactose will break down into glucose and galactose in presence of the enzyme beta galactosidase. Beta galactosidase is the name of the enzyme. Lactose is a substrate. It's a form of sugar. It's a carbohydrate. It will break down and form glucose and galactose, and both of them, both these molecules will act as the energy source for bacteria. So bacteria, they need a source of energy and they obtain from these molecules. So these substances, the glucose or these carbohydrates will provide them the energy. So lactose need to be, or it will break down, lactose will break down into glucose and galactose when beta galactosidase is present. It's the enzyme which breaks down lactose into glucose and galactose. E. coli synthesizes beta galactosidase only when lactose is available. Hope you can relate to what I said a few seconds back. See, this is the enzyme. This enzyme is needed for breaking down lactose. If lactose is not there, this enzyme is also not needed. Only when lactose is there, we need beta galactosidase. So in the absence of lactose, the enzyme need not be produced, or the genes coding for this enzyme need not get expressed. The genes coding for this enzyme should get expressed and form this enzyme only when lactose is there. Otherwise, it will be jobless. So there is no need for synthesizing it in the absence of lactose. Hydrolysis of lactose in E. coli. Lactose in presence of enzyme beta galactosidase will break down to glucose and galactose, which will provide or which will act as source of energy for bacteria. And this enzyme need to be synthesized only when lactose is present. Hope you are getting it. So that how the gene expression is regulated. When substrate is not there, we don't need an enzyme. Enzyme is a polypeptide. Enzyme is a protein. So the genes which are coding for this particular enzyme need not be expressed. And when lactose is there, when lactose is present, then we need to, or when at that time, these genes need to get expressed to form beta galactosidase. Are you getting it? Are you following?
Yes, ma'am.
Okay. Gene expression is regulated by controlling the rate of transcriptional initiation in prokaryotes. So if you take the case of prokaryotes, and children, I forgot to give one more example about the beta synthesis of beta galactosidase I told. Apart from that, another example is the development of embryo. Zygote will transform into embryo, and this embryo will get it will undergo differentiation and form an adult organism. So that is also this development and the differentiation of embryo is also under the control of gene expression. So many genes, a series of genes need to get expressed so as to bring about the development and differentiation of embryo. So there are different processes in our body which need to be, or for that we need to, or for all these processes, the expression of genes to form different proteins or polypeptides are very important. So what we discussed so far was about the gene expression in prokaryotes, and I told you the expression, gene expression is regulated at 4 levels: transcriptional level, translational level, and when it is being processed, say for example in splicing, or when the messenger RNA is getting transferred to cytoplasm from the nucleus. But when you take the case of prokaryotes, like bacteria, the gene expression is regulated mainly at one time, that is during transcriptional initiation or during the time of transcription, by controlling or regulating the initiation of transcription. You know that the transcription process should be initiated. Initiation, elongation, termination, all that you have learned. So by regulating, by controlling the rate at which transcriptional process is initiated, the gene expression is regulated in prokaryotes. So in prokaryotes, mainly the control of gene expression takes place by regulating the rate of transcriptional initiation, whereas in eukaryotes, I told you 4 ways by means of it, or 4 stages where they are being regulated. But here the regulation is mainly taking place at the time of transcriptional initiation. By regulating the rate at which the initiation takes place, the regulation, gene expression is regulated in prokaryotes. The activity of RNA polymerase. You know that for the process of transcription to start, RNA polymerase should bind with the initiation factor and they should go and get attached to the promoter. Only then the process begins. Hope you haven't forgotten. RNA polymerase, DNA dependent RNA polymerase will get associated with the initiation factor and they will go and bind to the promoter, and that is how the process starts. That is how initiation takes place. The activity of RNA polymerase at a given promoter is regulated by different accessory proteins. So there are accessory proteins which will regulate the activity of RNA polymerase at a given promoter. If this enzyme should bind with the promoter, we need the presence of certain proteins. Only with the help of this initiation factor is actually a protein. So here I told you that rate of gene expression, or the regulation of gene expression, takes place by regulating the rate of transcriptional initiation. For the transcription process to get initiated, the RNA polymerase should bind to promoter, and this is regulated by different accessory proteins. And accessory proteins can affect the ability of promoter to recognize the start sites. So if these proteins, these proteins, they can affect the ability of the promoter to recognize the initiation sites. So the promoter cannot recognize the initiation site, so the process has to start from promoter. So if there is a defect in the promoter, or if it is unable to recognize the initiation sites, the process, the transcription process cannot take place. So there the proteins, so these proteins regulate the ability of the promoter to recognize the start sites. And these proteins, you call them as regulatory proteins. They can act positively and negatively. when they are acting positively. Then when they are acting positively means they are promoting the transcription process. They are speeding up the rate of transcription process because they will enhance the ability of the promoter to recognize the initiation site. At the same time, they can prevent it. They can act as repressors. When they are acting as a repressor, they will prevent, they will affect the ability of the promoter to recognize the initiation sites, and thereby the process transcription cannot take place. Once again, I will repeat it. This was about the regulation of gene expression in prokaryotes. And here the regulation is taking place just at one stage mainly, that is at the transcriptional level. And this is taking place by regulating the rate at which transcriptional initiation takes place. And there are several proteins. And these proteins, we call them as regulatory proteins. And these regulatory proteins, they can act both positively or negatively. They can act as activators and regulators. And if these proteins are affecting negatively, then the promoter will not be able to recognize the initiation site and the process cannot take place. And if they are promoting, if they are acting as activators, it will speed up its ability to recognize the initiation sites and the process can proceed. The rate can be enhanced. The regulatory proteins and how this regulatory protein is affecting the transcriptional process. They will interact with specific sequences of DNA. They'll be interacting with specific sequences of DNA called operator. And these operator can regulate the accessibility of the promoter. You haven't learned earlier about operator. These are specific regions of DNA, specific sequences of DNA, and you call them as operators. Regulatory proteins, they will associate with certain specific DNA sequences. And what do you call these DNA sequences? Operators. You have learned about promoter, terminator, but not about operator. Regulatory proteins, they will interact with operators and thereby, that is how. So already I told you in the previous slide, the accessory proteins can affect, they can affect the ability of the promoter to recognize the start sites. How they are affecting? Explanation for that is in the next slide. I told there are certain specialized proteins. You call them as accessory proteins or regulatory proteins. They will affect the ability of the promoter to recognize the start sites. If they are affecting it negatively, you call them as repressors. Repressor proteins. If they are promoting it positively, they are known as activators. And how do they affect the ability of the promoter? How they are influencing? See, these regulatory proteins, they will interact with operators. What are operators? Specific sequences of DNA. They are lying close to the promoter. These sequences are lying close to the promoter and these sequences are known as operator. So the regulatory proteins will interact with regulatory proteins. They will interact with operators. And in this way, they will affect the ability of the promoter. Details you will learn. Once a regulatory protein is interacting with the promoter, it can interfere with the accessibility of promoter. Regulatory proteins, they can affect the ability of the promoter to recognize the initiation site. And how it is brought about? There are specific sequences in the DNA which you call as operator. These operators are lying close to the promoters and these promoter proteins will bind and this will accessibility of the RNA polymer not come and bind with in presence of regulatory proteins. When these regulatory proteins, when they are negatively affecting, you call it as repressor. When they act as a repressor, they will bind with operator. So when these regulatory proteins bind with operator, our RNA polymerase cannot bind with this promoter to start the process. Are you following? Okay. I'll repeat again. We were learning about regulation of gene expression. In prokaryotes, the regulation is taking place at the transcriptional level. How? By regulating the rate of transcriptional initiation. And you know that in a normal procedure, RNA polymerase will bind with the promoter with the help of some initiation factor, and the process will start. But there are certain proteins which can interfere with this process. You call these proteins as regulatory proteins or accessory proteins. They can act as repressor or as activator. When do you call them as a repressor? When they are inhibiting, retarding the process. And when they are promoting the initiation of transcription, you call it as activator. And what is the role of these proteins? How do they affect the process to, or how do they affect the transcriptional initiation? They will interact with special sequences of DNA. What do you call them? Operator. Where are these operators present? They are lying close to the promoter. And these repressor proteins, say for example, it is affecting it badly. Here you need to learn only about repressor proteins, not about activators. So say for example, this repressor protein is binding with operator. Once they bind with the operator, then our RNA polymerase cannot access the promoter to start the process. And that is how they are regulating the transcriptional process or transcriptional initiation. Usually, the operator sequences bind with a repressor protein. And once the operator or the repressor protein has got attached to the operator, then the RNA polymerase cannot bind with promoter to start the process. Here till here, have you understood? Have you understood, children?
Yes, ma'am.
Okay. Next, here it is mentioned each operon has got its own operator and repressor. So you should know what is meant by an operon. So operator and repressor, I hope you have understood these two terms. Operator means specific DNA sequences, sequence of a DNA or a particular segment of DNA. Where are they present? They are present close to the promoter. In the transcriptional unit, close to the promoter, you can see this operator. And these operators will be binding with repressors. What are repressors? They are some regulatory proteins which can interfere with the initiation of transcription. And these two will bind. Once the operator and the repressor joins together, then transcriptional process cannot start. Because if these two have joined together, then RNA polymerase will be unable to bind to the promoter to start the process. And each operon, now you should know what is meant by operon. Operon is an arrangement or a functional unit of DNA, containing a cluster of genes all under the control of a single promoter. When a single promoter is controlling the expression of a series of genes, you call it as operon. Another definition, I think the definition given in your textbook is an arrangement. Operon is an arrangement where a polycistronic gene. Polycistronic gene means a gene which codes for several polypeptides, more than one polypeptide, or for several proteins. An arrangement where a polycistronic gene is regulated by a common promoter, only a common promoter. One promoter is regulating a set of genes. Same promoter. It's a DNA sequence. A sequence of DNA where the promoter is regulating a set of genes. Once again, it's an arrangement where a polycystronic gene. Polycystronic gene is a gene which codes for several proteins or polypeptides. It's an arrangement where a polycystronic gene is regulated by a common promoter and regulatory genes. In a simple term, we can say it's a sequence of DNA where several genes are under the control of a single promoter. One more definition, a functional unit of DNA. Operon is a functional unit of DNA containing a group of genes or a cluster of genes under the control of a single promoter. One promoter influencing the expression of several genes. So each operon, there are different operons, Lac Operon, Trip Operon, Val Operon, so many operons. Lac Operon, Lac stands for lactose. Val Operon, just a minute. Expression of different genes. See, Lac Operon, it is the one which is regulating the expression of lactose, or the genes responsible for the formation of or conversion of lactose into glucose and galactose. There is a Lac Operon, Trip Operon. Trip stands for tryptophan. Trip Operon, Val, Valine Operon, His Operon for histamine. So many operons are there, and in each operon there will be an operator and a repressor. And what you need to learn in detail is Lac Operon. That can be asked. Used to be asked as an essay question, but you people are lucky. No essay questions. But you have to be so thorough with the concepts, only then you will be able to answer the MCQ questions. Each operon has its own specific operator and repressor. Lac Operon. Say for example, Lac Operon. Lac Operon is having Lac repressor, Lac operator. So it is specific to that. Lac Operon is having its own repressor and operator, and you call it as Lac operator and Lac repressor. Okay. So there are certain regulatory proteins which can influence the expression of genes, and these regulatory proteins can be either repressor or activator. Repressor means when they will retard the rate of transcriptional initiation, they are repressors. When they are promoting the formation of these proteins, promoting the formation or promoting the transcriptional initiation, then it is known as an activator. But here we need to learn only about repressor protein. And repressor protein, how they are affecting transcriptional process or how they are affecting the initiation of transcription. These repressor proteins will bind with operator. What are operators? They are specific sequences of DNA lying close to the promoter. And then what happens? Once they bind, once the repressor protein binds with operator, what will happen? RNA polymerase cannot access the promoter site. So without that, the process cannot proceed. Okay? Is it clear? And what you need to learn in detail is about Lac Operon. Operon is an arrangement where a polycystronic gene is regulated by a common promoter and regulatory genes, or else it's a functional unit of DNA where a cluster of genes is under the control of a single promoter. Lac Operon, Trip Operon, His Operon, Val Operon are the examples of operons. We will continue next class, children. The Lac Operon you will be learning in detail next period. But before you join, just read whether you have understood the concept. And if needed, I will repeat. And Nandana had asked me a doubt also regarding elongation. That also I will repeat next period. Okay, only elongation part, no? You have doubt. next session, I will begin with the recap of this particular topic. Okay, just read it and come. Thank you, children. See you next period.